All right, let's take a look at human physiology. We'll start with asexual reproduction. Asexual reproduction produces offspring that is genetically identical to the parent cell. This means that offspring can be created quickly. Asexual reproduction is advantageous when the environment is stable because the offspring are identical to parents. However, environment change could lead to the entire population being wiped out. Parthenogenesis is a natural form of asexual reproduction in which growth and development of embryos occur without fertilization. In animals, parthenogenesis means development of an embryo from an unfertilized egg cell. Cleavage is rapid mitotic division of the zygote that begins immediately after fertilization. Cleavage ends in the formation of the blastula, while gastrulation involves rearrangement of the blastula and begins with the formation of the blastopore. Gastrulation ends with the formation of the embryonic germ layers. These layers are the ectoderm, endoderm, and mesoderm. Let's take a look at those three. The ectoderm is one of the three primary germ layers in the very early embryo. The other two layers are the mesoderm, which is the middle layer, and the endoderm, which is the most proximal layer. The ectoderm is the most exterior layer. It emerges and originates from the outer layer of germ cells. Generally speaking, the ectoderm forms the nervous system, which is the spine, peripheral nerves, and brain, tooth enamel, and the epidermis, which is the outer part. It also forms the lining of the mouth, nostrils, sweat glands, hair, and nails. The mesoderm aids in the development of blood, bones, and muscle. Some of the mesoderm derivatives include the muscle, smooth, cardiac, and skeletal, the muscles of the tongue, the pharyngeal arches muscles of facial expressions, connective tissue, dermis, and layers of the skin, bone, and cartilage, blood vessels, red blood cells, white blood cells, the kidneys, and the adrenal cortex. The endoderm gives rise to the digestive system. Cells migrating inward along the archaeoteron form the inner layer of the gastrula, which develops into the endoderm. We also have the neural tube. The neural tube is the spinal cord and thus also the nervous system. The neural tube is the embryo's precursor to the central nervous system, which comprises the brain and spinal cord. The neural groove gradually deepens as the neural folds become elevated, and ultimately the folds meet and coalesce in the middle line and convert the groove into the closed neural tube. In humans, neural tube closure usually occurs by the 4th week of pregnancy. Organogenesis is the process by which the ectoderm, endoderm, and mesoderm develop into the internal organs of the organism. This means that cells are continuing to develop. An embryo is divided into two hemispheres: the animal pole and the vegetal pole within a blastula. The animal pole consists of small cells that divide rapidly, in contrast with the vegetal pole below it. The animal pole is the top half of the embryo and is where most cleavage occurs. Remember that cleavage is rapid mitotic division of the zygote that begins immediately after fertilization. The vegetal pole is the bottom half of the embryo, is yolky, and very little cleavage occurs there. Apoptosis is a process of programmed cell death that occurs in multicellular organisms. Biochemical events lead to characteristic cell changes and death. Triggering signals can come from the nucleus, the endoplasmic reticulum, from outside the cell, and the mitochondria can initiate apoptosis by sending a relay protein to the cytoplasm. It is estimated that between 50 and 70 billion cells die each day due to apoptosis in the average human adult. In contrast to necrosis, which is a form of traumatic cell death that results from acute cellular injury, apoptosis is a highly regulated and controlled process that leads to advantages during an organism's life cycle. Homeotic genes are genes which regulate the development of anatomical structures in various organisms such as insects, mammals, and plants. There are several subsets of homeotic genes. They include many of the Hox and parahox genes that are important for segmentation. Hox genes are found in bilaterian animals, including Drosophila and humans. Hox genes are a subset of the homeobox genes. The Hox genes are often conserved across species, so some of the Hox genes of Drosophila are homologous to those in humans. Despite the terms being commonly interchanged, not all homeotic genes are Hox genes. The MADS-Box genes are homeotic, but not Hox genes. Thus, the Hox genes are a subset of homeotic genes.